Hello, this is Dr. D, and today we are going to talk about blood type. All right. Now, you've heard of the types of blood A, B, and O. All right. And you've often wondered, what does it mean? Well, what it means is that the type A blood has a special molecule on its surface which are called antigens. These are cell surface molecules on the RBC. These can be a protein, a fat, a carbohydrate, a sugar, which are recognized by the immune system. So antigen you can think of is a marker. All right, but they have to be it's a very specific marker. All right, so here the best part is that antigens are recognized by antibodies. So think of a lock and key. Just like a lock can open a particular a key can open a particular lock and binds, same thing, an antigen and an antibody are very specific. So in general, what are antibodies? These are immunoglobulins. Remember, we talked about the blood plasma contain protein called globulin. Okay, so antibodies are the immunoglobulin proteins that bind to specific antigen, not any. Just like your car key cannot open my car door, just like that, a specific antibody will bind to a specific antigen. All right. So ABO blood type is dependent upon the antigen. All right, let's see this slide so you'll understand better. All right, see this slide. This is type A blood. So what's type A blood? Type A blood, as you can see, the RBC has this antigen, this molecule, this specific molecule called A. All right, and type A blood will have an antibody, and think of antibody as a soldier. So this antibody is going to target what? B. Obviously will not target A, otherwise this your own blood, the soldiers are going to attack your RBC. Are you understanding? So type A blood has this specific marker A, and it has a soldier that will target B. Now, if this type of blood is mixed with a blood B type of blood, and a B type of blood on the RBC will have another antigen called what? B, right? But it will have a soldier that will attack what? A. So if these two bloods are mixed, you will see agglutination will take place. Clumping will take place. Will that be of any use? No, that will be damage, right? So here, blood type A contains antigen what? A. And it has the soldier that will target for B, right? So it's called anti-B. B has the antigen B, and antibody, what does it have? Anti-A. AB now has the RBC, which contains both A and B. But can it afford to have a soldier that will target A and B? Answer is no. It will attack its own RBC. So it neither has antibody A or antibody B. O type of blood has an RBC which does not have A or B antigen. All right. So it will have the soldiers that will target both anti-A and anti-B. So O obviously are considered universal donors. Why are they considered universal donors? Because look at their RBC. Their RBC has no antigen A or antigen B. All right. It doesn't have those markers. So it can be accepted by A or B, right? And why are AB called universal acceptors? AB are called universal acceptors because do they have soldiers that will target A or B? No. So they can take blood from A and they can take blood from what? B. All right. Then we come to another antigen, which is called the Rh antigen. And it is called Rh. Why? Because it was found in the rhesus monkey. Later on, what they said was, or what they found was, that this particular antigen is also found in human beings and it is very widespread. But the Rh stayed. So if your RBC, it doesn't matter whether you have A, B, or O, or AB, if your RBC has the Rh antigen, you are called positive. If you don't have, it is called negative. Now, why is this important? Why is this Rh so important? It is important, especially if a Rh female marries an Rh male and carries a baby. So when the baby by default will be Rh positive. All right, because Rh positive is a dominant characteristic. It is always going to show up. So because the baby is Rh positive, the fetus, the mother is going to start producing what? The mother's, because of the mixing of the blood, right? There's some amount of mixing of the blood between the fetus and the mother. So what happens is that the mother will start building up Rh positive, antibodies to attack the fetus. Do you understand? And what this is called the mother's body is going to get sensitized. It is going to have antibodies that is going to attack anything with Rh positive blood cells. So the first pregnancy already is going to be hard, but the second pregnancy will be impossible. Why? Because now the body has become sensitized. What does it mean? She has antibodies that will attack anything that contains or carries Rh positive antigen. So the fetus, as long as she's married to the same man, to Rh positive man, she's going to carry an Rh positive fetus. So now in the second pregnancy, the antibodies that she has built is going to attack the fetus. Do we understand? So here, if what happens is initial transfusion is okay, but later on lead to what? Blood agglutination, clumping. I showed you clumping, right? This one. And either it will lead to a miscarriage, or the baby is going to be born with huge amount of issues. All right. So the way the doctors, they regulate this is the mother is going to have to take RhoGAM shots, and these will intercept the incoming Rh cells from the fetus. Are you understanding? So the Rh cells are not going to, it's going to interfere, stop them, and it will not allow the mother to attack the fetus. All right, with this we come to end of our presentation. So if there are questions or concerns, please stop by. And I thank you for listening to my presentation. Have a great day.